Yeah.
Okay, we're starting. Okay. Okay, we're gonna first start about chapter 16, which revolves around DNA structure and replication. Okay.
Hey, Matt, you coming tomorrow?
Yes.
Dude, that's a big W then because.
Hey, yo,
Nathan,
whoever
you
got
wanna
the
come
echo,
too?
I'm muting you right now. Oh,
What
that's
was that?
Corey.
Me?
What'd you ask, Corey?
You wanna come over tomorrow?
I want to come over to what?
His
I
house.
said, you wanna come tomorrow?
To what?
We're studying for bio.
Wait, what time
Oh.
tomorrow?
What time?
Time.
Huh? Same time?
Same time.
Same time.
Okay.
Like
At
7:30.
the where?
Corey's
My
house.
house.
Oh, yeah, sure. Wait. Yeah, sure.
What do you call it though, Brian? You
Are
guys
you
gotta
going to
bring
have
some.
your shirt off?
Hey, yo.
I'm gonna have all my clothes off.
Okay.
Oh,
Bro.
perfect.
Okay, we're gonna now officially start. Okay. Okay, we're gonna first start with DNA structure and replication. DNA is like, do you know why DNA is so important? Is because like it's there to like do specific jobs, and we need to replicate it to grow in that stuff. Okay, so we're gonna first start off with the structure of DNA. Okay, so I'm gonna draw this out. Where is it? No. There we go. Okay, DNA structure is bop 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 bop. Oh, bop 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 bop. With a do 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 do. This is very scuffed, but it will make sense. That's 5 sides. Okay, just imagine.
Should we copy that?
Nah, you don't have to. This
Is
is
it a
just
good idea
this is just a scuffed version. Okay. So
Matthew, are you recording?
yes. So, oh, and also for the basis of DNA, the main distinction from DNA and RNA is that DNA has 2 strands and RNA has 1 strand, and DNA is the recipe. It's the master recipe. It's the master chef. Okay, it's the master cookbook.
Chef.
It's the master cookbook. Well, RNA is the messenger. It provides these recipes to everywhere else. Okay. And
Is it
also,
like the
DNA
waiter?
is DNA
No?
is 2 strands,
Okay.
and RNA is 1 strand. Okay. So I'm gonna type that out. Oh no, I'm a buffoon. Okay.
No, you're not.
RNA
You're Matthew.
RNA is equivalent to messenger and 1 strand.
You should show it to Mr. Wayne that you do this, and then he'll probably like be like,
Nah.
"Good job."
Okay. So there's a few things you have to know about the structure of DNA. Okay. So the first thing is these circles. These circles are phosphate groups. Okay. So phosphate groups. There's a phosphate group, and there's a 5-carbon sugar. Okay, and and the link and when there's a 5 and from the bond it takes from the 5-carbon sugar to the phosphate group. It's called what you call? Oh, it's called the phosphodester bond. Okay, phosphate group phosphodester. No, phosphodester bond. Okay, I don't know how to say it, but once you see on the test, click this in. And it's a covalent bond, which means it's a very strong bond, which holds the phosphate group. Phosphate group and 5C sugar. Okay, so that's important as it is important because in order. Oh wait, in order for it to, we need the phosphate group and the 5-carbon sugar to stay strong together, which is why it's a covalent bond. Okay, so. Oh no, that's fine. Okay. So we know three of the main components, okay? But there's also these things in the middle. These things are nucleotide. Wait, is it They're not nucleotide. Oh no, they're nitronous bases. That's what they're. They're nitronous bases, and these, just imagine they're just a way to connect things and show and represent each distinctive genetic code for each person. So, let me see. Okay, so we know that these are nitronous bases, and between the things that hold these bases together are hydrogen bonds. And the main reason why it's hydrogen bonds is because you want it to break away. And the reason why you want to break these bonds is so you can do replication, which we'll further establish later on. Okay? Hydrogen bonds for nitrogenous bases. Okay?
Tim, we can hear everything.
Okay. So you're not alright? And what you also should remember is that A and G are called purines, while C and T are pyramine. Okay, and what you remember, and also A to T, the relationship is A will always link up to C, and G will always link up with C and C. Okay, so A and G are pyrines, and C and T are pyridines. Just remember that. There's no like real specific reason why they're like called that. It's just remember that they're called that. Cause like if it pops in the test, it's just you have a general idea. Okay.
Did Mr. Wang ever talk about uracil?
Uracil? Uracil's in the RNA. I think it replaces.
It replaces.
What does it replace?
It replaces T.
Okay, yeah. So uracil will be uracil will be a
I don't know if
pyridine.
he talked about it.
He didn't really talk about it, but in that situation, then if you know that it replaces T, it would just mean it's a pyramine. Yes. Okay. Okay. So the reason why DNA is called semi-conservative is the fact that every time it replicates, it will always have an old strand. Okay. DNA replication.
What do you mean old strand?
So in DNA replication, they have the original strand. They have two strands, right? They unzip each strand, and then that strand is by itself. So there's only one strand on each side, right? So I'll just draw this out. Okay. Oh no, wait.
Oh, please, thank you.
No, it's fine. Wait, how do I go back?
Undo.
Oh, that's fine. I'll write that later. Oh, I need to type. Yeah, I'll type this out right now. Semi-conservative, because will always contain an old strand. Okay. So in DNA replication, you have these two lines, right? Okay, Tim.
Yes. Following.
Okay.
Yes.
When a DNA replicates, they split these two lines, these two strands, into separate sections, right? And this strand gets replicated to make another strand, so it combines into another DNA. But as this is always constant, right?
Mm-hmm.
This applies to this fact as each time it replicates, there's always one constant strand that's always there. Cause like it's just a continuous cycle, okay?
So where do all these extra ones go?
What do you mean extra ones?
Oh, the old strand.
No. What I'm trying to say is that every time it unzips that strand. So for example, that turns into this, right?
Hmm.
But then you unzip it again. There's still this old strand. It's still there.
Okay.
Wait,
So
so like,
What is the purpose of that strand?
It's just there. No, cause like there's no real purpose. It's just there for it's a template that helps further bond other nitronous bases or like the other strand to them. So like it's just a continuous cycle of them.
So if the one that you circled on the bottom left were to replicate, there would be another strand left.
Yeah, cause when this gets replicated, these two strands go away again. They separate again, and then they form another. They form their own respective strands for DNA.
Yeah. Okay, got it. So they're just there. There's
Yeah.
no real.
They're
Okay.
just their main importance is that they constantly cycle. They constantly cycle, keep on going. And when they use DNA replication, okay?
Okay.
Oh, okay. I'll record this, so I'm just gonna delete this part. I'm just gonna skadoosh it. Oh, where's the eraser? And destroy that.
I'm gonna talk about how DNA is anti-parallel.
Oh, yeah, I'll get to that. I'll connect it to that later. Okay. That's a
On
good
part
question.
3.
So we're now gonna talk
Close the
about.
door.
We're gonna talk about replication fork, origin of replication, and all that. Okay. So we're gonna first start with origin of replication. Right, I'm just gonna draw a diagram. It's gonna be like this. Oh, where's the pencil? There we go. What? Okay, origin of replication will always be in the middle. Okay? And what's so important about origin replication is that it's where DNA replication begins. It's where it starts, okay? It's where it starts. Okay. And in this replication, there's two strands. There's the leading strand and there's a lagging strand. So the leading strand will always be okay. Oh, okay. Wait. Give me a sec. I need to draw the three ends out. The three goes from. Oh yeah. So goes there. Give me a yeah. This is 5N, 3N, 3N, 5N. And this is where Nathan's question of anti-parallel comes from.
Mm.
'Cause- no, I'm a buffoon. There we go. So anti-parallel is just basically parallel. I can't spell, but this is fine. Yet opposites. Okay? Like not really opposites, but it's flipped. So basically, they're parallel to each other, right, Nathan?
Yes.
But they're
Can't
also
you just say that like the side that has like the 5 end, the other side will have the 3 ends?
Yeah,
No matter what.
that's basically it. Yeah, so basically they're just parallel to each other, but they're basically opposites. One is the backwards and one is the otherwards. And what you also gotta remember is that the leading strand will always go towards the 5 end. Okay? So this is the leading strand, and it's going towards the 5 end. But in the other strand below, it will go towards that way. So it'll look like this. What? That's the leading strand, okay? So the leading strand the leading strand will always permanently elongate. It's just a one continuous line, okay? It's a one continuous line that goes along with the DNA, okay? So this strand is just constant. It's always growing, okay? Just remember that. It's constantly growing. And it'll constantly grow to make another strand for DNA replication. As technically after it's done, it will be shown as two different well, give me a sec. I'll draw the result right now so you can see what it should look like at the end. What page is this? 304. No, come on, load. Bio textbook. Yes, okay. At the end, it'll be like this. There's two DNA sections, okay? So, these leading strands, they're constantly elongated. But what about how about the space down here? That's where lagging strands come in. Okay? So lagging strands are these small fragments. They're basically these small sections that later on get fused together into one long into the everything at the end, okay? So they get fused together at the end. So, and within these fragments, there's also these Okazaki fragments that are just basically a type of lagging strand. There are also segments that get later on fused together into one big one. Okay. So the main takeaway here is that. Oh, and also I forgot to say this. Alright. Origin of replication. This is where DNA replication begins. That's what we're going to talk about right now. Replication fork. I'll further elaborate that. End of each replication bubble where new are formed. Leading strand elongates. I can't spell, but it's fine. Permanently, you get what I'm trying to say. Permanently, and goes along within DNA lagging strand. Small sections that get synthesized into a bigger one. No. Haha. Okay. So, okay. Replication fork is this line right here. And when I wrote the definition, just basically at the end of each replication, there's this little kind of subsection, right? And as the lagging strand and leading strand further on and get fused, they get fused with this replication fork in order to form this new DNA strand. Okay? So the main takeaway is that there's these bubbles, right? And these bubbles are where the system of this new strand gets formed. Okay? There's the replication fork, origin of replication, leading strand, lagging strand, and Okazaki fragments are just basically a subsection of the lagging strand that gets fused together. Okay. Now we go to. Did you guys write this?
Matthew, Corey's got something to say.
Yes, Corey, you can just unmute yourself.
No, you literally muted me, and I can unmute.
Yes, that's you deserve that. I'm joking. Okay, what do you want?
Okay, wait. What color is the Okazaki fragment?
Oh, I didn't draw it out yet. I just told you definition.
Okay, so
It's a lagging strand, right?
Yeah, it's part of the
So-
lagging. I'll further draw. I'll draw it in like the next section that I'm gonna talk about in like 2 minutes.
Wait,
Wait, wait.
are those lines that you drew, Matthew? Like what it's gonna end up being, or
Yeah,
what
yeah.
it's
Because like you see how there's these lagging strands and leading strands of replication fork up top of like right here. This also is applied to down here too, which leads to two DNAs forming.
So wait, okay, so the
Okay.
lagging
Oh,
strand.
okay.
Because you see how like
Fine, right?
yeah, the lagging strand, leading strand, and replication fork are later formed together to make another strand, and we'll go to specifics next in like the next
okay,
minute.
the lagging strand is the red thing, right? Like
Lagging,
the
no,
red
the
arrow.
lagging strand is the blue thing. I mean, the lagging strand is the red thing. Yes, this is the lagging strand. This one.
And then the leading is the blue.
Yes, and this right here is the replication fork.
Okay.
Wait, Matthew, can you like briefly go over like the differences in the
Oh,
strands and
wait.
the main point of like why is it doing this?
Oh, that's what I'm gonna go specifically later in the next,
Okay.
next few minutes. Oh, and also, you can say it's kind of. You can say that there. You can also say the definition of derived or lagging is the fact that DNA is supposed to start right here, and it's just basically lagging behind the leading.
Also, are you recording this, Matthew?
Know that?
Yes.
Okay.
Do we have to know that?
No,
or...
but like that's just literally the name.
Okay. Well. Okay.
Okay, we're gonna.
Okay.
Oh no,
Yeah.
I'm gonna go next now. Okay.
Did you go next?
New. Don't save. Okay, now we're gonna go to specifics. What? This is not gonna be fun. Okay. Okay, we're gonna go specifics in that situation where we saw that bubbles and all that, right? So I'm not gonna draw what specifically happens in here. These things are separate DNA. Remember that, okay? So
That is DNA being unzipped.
this is this is being DNA unzipped.
Unzip your pants.
What?
Okay.
Corey says, bro.
This is DNA being unzipped. Okay.
Okay.
And you know that there's one side is 5n and the other side is 3n. And due to anti-parallel, the other side must be 3.
3 to 5. Wow.
But what you also gotta remember is that when
Wait, Matthew. If I'm gonna color code this, what should I do? Or
you should color you should make this the original DNA segment the both sides the same color.
Mhm. Okay. And then everything else you'll just say.
Yeah, everything
much.
else I say. Okay.
Okay.
So we have this 5n and 3n, right? And what you gotta remember is that what you call it. Oh yeah. So at this at this point, that's where the origin of replication is, okay? Because like that's where DNA starts and ends. Like that's where it starts, but also where it can end, okay? Wait. Actually, nah. This is fine. This is fine. Oh, if you guys have your textbook, you can open to page 304 in the original textbook. Or
Last is open to page 164.
304.
304.
Yes, and this is it gives what you call it? It gives a more in depth. Or like I'm just basically using that template. Okay. So you know there's a 3n here, 5n here, 5n here, 3n here, okay? Yeah.
Wait, you said 304?
Yeah, 304 to get. I'm just
Yeah,
I'm
304 is
basically.
a chi-square.
Oh, I'm talking about the original textbook.
Oh,
The red one.
Oh, okay.
Just try. It's just search up the chapter that we're on cell cycle. We're on DNA replication, all that. Nathan. Try to find this.
Yeah.
There should be a same thing in the new textbook too. Okay. So first things first, we have this big blob in the middle right here where it starts, and this is helicase. And what helicase's function is, it unzips the DNA. Okay? I'll move this up here. Helicase. No! No! You buffoon. Get out of my... There we go. Okay. Helicase. Enzyme unzips DNA. Okay? So we have helicase. It unzips DNA. So it basically separates them, okay? So the bottom here, this strand is at the bottom, okay? It's at the bottom. This one's on this basically just infer this one's on top and this one's at the bottom. Okay? So
Wait, what does that circle represent?
helicase. It unzips the DNA strand, okay?
Okay, just making sure. Alright, thank you.
Okay. This one at the bottom, there that's where we're gonna factor in this one at the bottom. That one, it is primase, okay? And if you have the old textbook, you can see what primase's function on the top table. But I can also say right now. So primase's function for the lagging strand, which is gonna be on the bottom here, because if you look on the replication fork, the top in this part, the top right here will be the lagging strand, and the bottom, the leading strand, and the bottom right here is the lagging strand. Okay, I'm gonna draw the little dots of the lagging strand. Okay, And what you gotta remember is that so for the leading and lagging strand, I'll give a specific color. So lagging strands are these subsections, right? They're not one long thing. They're just these subsections. Just remember that.
Mm.
Wow, this one on top is one continuous leading strand. And what you gotta remember is that it will always go towards 5N. So it goes in this direction, okay? And this goes in that direction, okay?
Okay, so 3 to 5 and then 3 to 5.
It always goes to the 5 end, okay?
Okay.
Okay, so you know that these small things are the lagging trend. These things, this big thing is the leading trend. So what primates does, which is so important, is that as you can see, if you have the old textbook, you can see the differences. But specifically for lagging strand, it synthesizes the 5 end to each Okizaka fragment. So as you know before, each Okizaka fragment is the subsection of the lagging strands, and what it does is it synthesizes it to the 5 end. Synthesizes at the 5 end. So like at the end, it helps at the end synthesize all of it together to one big line. One big strand. Okay?
Can you spell whatever the fuck, please?
Yes. I will. What in a regular case? Oh, and just remember, everything that ends with an ace is an enzyme. DNA strand. Primer. Wait! Give me a sec. I'll just call. I found this in the textbook. Oh, wait. What you call it? Tim.
Yes.
Send me a picture. You have the old textbook, right?
Don't. I looked at it's like.
Jeffrey, do you have yours at home?
I have it.
Actually, either of you guys. On 304, can you just send me a picture of that table?
I got you, Matthew.
There you
Go, Corey, bro.
I'll just copy and paste that. I'll just
Let's
copy
go,
and paste
Corey.
that.
Let's go.
Let's go.
Like I be telling.
Okay, but how do you spell Okin?
You literally watch an anime. You should know this. You watch an anime.
I'm sorry. I don't know how to spell Japanese
It's
in
Okazaki.
English.
Okazaki fragment.
Wait, say it again.
Ozaki.
O-Z-A.
No, send it through Discord, Corey, you buffoon!
Ozaki. Ozaki.
Oh, okay.
Oh, okay, that makes a lot more sense. I thought Matthew said Okinatsu.
Okaizaki. Corey, is it uploading?
Yeah, sending it.
Okay. Everything's waiting on you, Corey. This is your fault.
Bro. Okay.
The later, the
joking.
longer it takes for Corey to send, the later I go to sleep, bro.
That's what I'm saying.
Bro, you sleep so early.
I wake up early.
You
At
still
5
lick
o'clock.
the business
Aha!
plan.
Huh?
No,
I
that
don't
is so.
sleep early, bro. I sleep between 12 and like.
Okay. This is kind of scuff, but you can kind of. I'll just
I
read it
wake
out
up
for
at
you.
5.
What the hell?
No way.
Oh, do
Oh,
you
Wait,
want the
look at 5.
That is so scuffed.
Huh?
This is
do you
fine.
want the top, Matthew?
Huh?
Oh, here, I'll send you a better picture.
Yeah, send me a bigger one. Send me a better one.
Damn. Okay, so let's see what that says. It says
No, we're not. Send me, Corey, please. Just send me the top half thing.
we are timeless. We
What?
are timeless.
After you turn on your camera.
Me? Oh wait,
Matthew, just use the one that I sent you.
Oh wait, that's so smart. That's what I'm saying.
That's the one from the newer one.
What, Tim?
Oh
Just
wait,
turn
that
your
Corey got
camera
me a good one. Oh, that's the
Yeah.
wrong one. Oh no. Whoa. There we go.
It's about drive. It's about power. We stay hungry. we stay alive. Put in the
No,
work.
why is it so
Yeah.
big?
That's what. Never mind. Never mind.
No.
Where's that from?
No. It's fine. I'll just explain it then. This is too much work. So basically, you can just write this down. Because
What is the blue thing?
How do you like case? Okay, just write this. I'll just write this.
No. The going to the 5 direction.
Which one? This thing?
The
Those are
Yeah.
This is the leading train.
laggers. Is that leading?
The top one is leading.
Mine's
The
lagging.
bottom one is
No,
lagging.
yeah.
Oh, I'll just type this out. This is too much effort to draw and everything. That is
Which
factual.
one you
So
talking
Primate
about?
says what synthesizes Ozaki fragments to strands?
I thought it makes RNA.
I don't know. Matthew said something.
No,
This is something that
Jeffrey, what you
synthesizes.
call it? It synthesizes RNA primer. It doesn't make RNA. I mean, it
Yeah,
doesn't make
Oh.
RNA primer. It synthesizes it. That's what it does.
Jonah, I'm
Yeah,
winning.
it makes RNA, right?
Not winning.
Let me search it up. Does
I'm
Primus
winning
make
in the
RNA
poll.
primer?
Remember the poll?
I know
think
you're
it
not winning.
does.
I am. It's 54 to
Yeah,
40.
or that RNA primer is RNA.
Bro, on my poll,
This
I'm winning.
synthesizes RNA primer.
Wait, Jonah, which one? Wait, Jonah, which one are you for?
He
Sour
wants
cream and onion.
dude. It's
And then
definitely
Corey is for
salt
Solomon.
and vinegar, bro. It's
Yeah.
literally
No,
salt and vinegar.
literally, it's me versus everyone else at the study session.
Same thing.
Fucking
I mean, sour cream and onion.
It's
It's
all
like
the same.
it's more like flavor.
Wait, does it?
Salt and vinegar is kind of
Let's
basic.
see. Actually, it says it makes sense how it synthesizes, but
My love will always be
Hmm.
this.
let me see. Primase. DNA. The primer is a short stretch of RNA with an available 3N. An enzyme called primase can start an RNA chain from scratch. I can see what you mean. Okay, so basically starts at the scratch. Okay, so yeah. So technically, you can say that it creates RNA.
Wait, what creates your RNA?
Primase, because it's
Nice.
able to start the RNA chain.
The primate starts.
RNA chain. So basically, what it's just trying to say that there's a chain that gets involved when creating these strands that helps that. Okay. So what you call it? They only can. This is okay. This is really important.
Is
Let's
that
write
right?
this down. You can only add nucleotides for the strand, but can't start it. That's
Wait, what's this? What's this part for?
no. This is just important overall.
Oh, okay.
You can't start it. That's where primase comes in and starts a RNA chain. Okay, this is correct. Okay, so basically, you see them adding these nucleotides, right, to the lagging strand and the leading strand, right? But they can't start it. You gotta remember that they can't start it. They only can add them. That's where primase comes in. So primase synthesizes RNA primer and able to start a chain. It's able to start the adding of nucleotides into the strands. Okay? So I'm gonna write this down. I'm gonna make a chart. Not really chart, I'm just gonna say boy. Okay.
Chasing the beat of my heart.
So primase, that's one important thing. If this primase is
the beat
the
of
table.
my heart.
Primase, leading strand. Yeah, this is why I don't like DNA replication because it's scuffed. DNA strand synthesizes.
Only ending.
Matthew, can you make the font a little bit bigger?
Yeah, I'll make it bigger.
Thank you.
Where's your house again, Corey?
My address is 950 Vista Grande.
Oh, awesome.
Do you need a ride?
No, I'm your neighbor.
No, you're not.
Nathan, bro.
Nathan, I know you live. Okay, that sounds scary, but I know where you live, Nathan.
Okay, wait.
No! That's fine.
I live near. If you know where Dylan lives, that's where I live.
Where you live?
Okay.
Why are there two terms?
Okay, for lagging strand.
Oh yeah, wait, no, you live there.
Yeah.
Oh yeah, wait.
The place with the whack streets.
Yeah, you live on that hill.
Yeah, no one really knows where it is.
I
Oh, I
do.
know where it is.
I know where it is. I was there today.
Your house is blurred out. What the heck?
Yeah, I know you can't see it.
Hey Julian. Hey bud.
What's up?
Okay.
I can't tell you how it all happened.
When is it again, Corey?
Tomorrow.
Bro, I made this hella fire
No way.
fucking playlist of just old Disney songs. And by old Disney songs, I mean just all Austin Moon. So I've been listening to that for like the past 2 hours, bro. Oh no, Heartbeat.
Oh, it's a W, bro. Matthew's coming tomorrow and Clea's coming tomorrow.
Oh wait, Kalia's gonna be there.
Yeah.
All right, not going. Peace out. Let's look. I'm controlling light. The light is in my hand, bro. Can y'all do this?
Look,
I feel like if we get off, no, this is a good thing.
What are you talking about?
Because if we get off task, Kalia is gonna make us get back on task.
Yeah.
Wait, who else is going? Is Justin
Me.
going?
I'm
I gotta
going.
work.
Oh,
Oh,
All
okay.
the people from today, basically.
I kind of want to.
yeah.
I'm gonna see if someone can take part of my.
Y'all control light like me though.
oh, you're funny.
Oh, actually, you guys do whatever you want right now. I'm just writing this stuff down.
I can do that too. Let me go get my flashlight real quick. Jeez. Many reasons I think about you every
Which
morning.
is 7 minutes in.
when
I
turn
feel
out
very
the
comfortable.
I just. Can we watch that video later?
What is it? Just tell me.
Thing? It's relevant to like recessive and stuff.
light. Okazaki.
Oh, why did Mr. Wang show us those videos about like animals reproducing? Like, what was
It's
it?
helpful
What
for our
was it?
learning.
Fire.
It's helpful
Hella
for our
cool,
learning to
what?
see how dolphins.
Bro,
Bro, why would you, bro?
bro,
Tim, didn't you watch them on your own time after school?
Yeah.
No, we watch them in class.
No, no, the
No,
dolphin one.
Tim watched more.
What's the
Right.
dolphin one?
Where's the dolphin one?
I watched the dolphin one in
They
class,
did.
bro.
That's
That
very
shit was-
educational, thank you.
What the fuck?
No, it's actually. I think it's pretty relevant.
Hairless monkey.
You guys like
Matthew,
these? You
wait, Matthew, what's DNA pull 3 and why is it before 1?
guys like these?
It's kind of stupid, cause like
What?
actually it makes sense
They're
why it's
okay,
before 1,
Timothy.
but it's also like it's one of those things how 3 is before 1.
But why is there no 2?
There's just no 2 of Julian. This is
There's
what,
just.
there's just no 2. It's one of
What?
those situations. There's just no 2.
What are you talking about right now?
The polymerase.
Yeah,
It's-
I'm gonna
it's
further explain this
3
after I'm done
before-
running everything.
It's 3 before 1, and then there's no 2.
Yeah, this is why
Because
science is
scientists were
stupid.
just on something else.
Okay,
Wait, one is
I'm
only on
almost
the
done.
lagging strand, right?
Yeah.
It removes the RNA.
Okay, what you call it? So you first start off with your helicase, your boy. Okay, now I'm gonna draw this out. Okay, this is gonna make sense now.
Boy.
Your boy helicase coming in clutch. You start with your boy helicase, and helicase does this thing where it unwinds stuff and just separates them. Okay, nice. Everything's fun and dandy. Very nice. Wait, 5N. There we go. Okay, your boy handy dandy. Then Primace comes in. Okay, Primace. Remember that Primace is for both, but in this situation, Primace is like more present, like in the first part for the lagging strand than the leading strand. Okay, so as I wrote before, Primace for the lagging strand synthesizes our primer at the 5 end of each Okazaki fragment, and it's further combined to the idea that Primace starts this RNA chain. So, and you know that you're trying to go to the right for the lagging strand. So that's where they start. They want to start there and they move towards there to the 5N. So they move towards there, okay? And that's why you don't really see a Primace up here. It's like back there. It's like the back spot, like around here basically. Okay? So your boy Primus creates this RNA primer chain and starts this strand for each respective leading and lagging. Okay? Then DNA polymerase 3 comes in. Let's make it orange. Yes, orange. So DNA's polymerase 3, it says continually synthesizes the leading strand adding onto the strand. So and since the fact that leading strand itself constantly elongates, there's no subsection. It's constantly growing. So the DNA polymerase 3 allows the leading strand to constantly go and keeps on growing. But since you know that leading strands are these subsections and Okazaki fragments are also these subsections of these small strands, DNA polymerase 3 in the leading strands it not only elongates to the Okazaki fragments, but also adds it to this one to the end where it's able to make this one gigantic strand for the lagging strands at the end. So basically, DNA polymerase 3 helps leading strand become bigger and bigger, while DNA polymerase 3 for lagging strands elongates and makes the Okazaki fragments larger and longer. Okay, so DNA polymerase 3 for both of them removes the primer at the end right here, right, and replaces it with DNA. So after it's getting close to being done forming a new strand, DNA polymerase 3 says no. DNA polymerase 1 says no, be gone. So then this gets destroyed basically. Houdini. Gone.
Houdini.
So and then it leaves this small section left over, and that's what gets replaced with DNA. Okay, that's where it gets replaced with DNA. So like it's connected, and then DNA's ligase 3 is able to join the 3' end, which the primer gets absolutely destroyed from the DNA polymerase 3, and then it replaces it with the rest of the leading strand. Okay. So basically, at the end, it will go from this primer, right here, primer, and then at the end, it becomes this. Strand. And then, yeah, so there's no primus and everything. It just becomes this one separate thing. Entity.
So what is the blue part?
This thing?
Oh, on the others, on the that.
Oh, that's basically the 5. This is that part. So I need to. Oh yeah, and also this is the 5, 3, 5, and this is 3, 5, 3. So at the result is basically it results in these two separate things, and that's the specifics. And if we're going to Corey's house tomorrow, I'ma rinse and repeat this again. I'm saying this again.
I got the whiteboard ready.
I need a big one.
Yeah, Corey. Woo!
No, I'm joking. It's fine.
Ooh, Corey.
Okay. Now we know that.
I'm over a bigger
We know
one, Matthew.
anti-parallel.
Wait,
No,
Matthew,
I'm
Matthew.
joking.
There,
No need.
which one's the original? The blue or the red one?
The original is the blue one.
It'd be the blue ones. Okay.
That's what I was
So
asking.
that's the old strand, right?
No. Blue. Yeah, the blue one's old strand. Yeah, the blue one's old one. That's what Julian's trying to say.
Yeah, that's what I meant.
Ooh.
You know what I meant.
Oh.
Wait, Matthew, can you like quickly go over what the Okazaki fragment is again?
It's just basically a subsection of the lagging strand that's just there to help out. It's just there. Cause like for example, DNA polymerase 1, DNA polymerase 3, it helps further elongate that lagging strand, which is the Okazaki fragment.
Oh, okay.
Yeah,
you.
it's basically really similar
Wait,
to
what?
each other.
What does it do again? Say that again.
It elongates the Okazaki fragment, so it basically just makes the strand longer.
What does?
DNA polymerase 1, right here. It
Jesus
says right here.
Christ.
Oh no! I got.
What do you mean? What do you mean, Tim?
Nothing, I didn't hear it.
You're so silly.
Wait, and Matthew, when it's getting like made, right? In the thing you drew, it goes from 3 to 5 and 3 and like that. That's how it builds.
Yeah, so basically, if the top part, the old one is 5 to 3, it will build in to the left. It will go in the left direction. So I'll just draw the arrow so it makes sense. Okay?
That's how it was built.
That's how it builds. It goes down to the left, and just imagine there's an imaginary primer right here, right?
Mhm.
Like right here. This gets destroyed this gets removed at the end. So like just remember that it gets removed at the end, okay?
Okay.
Okay.
Fuck, it's an eraser piece.
So
Oh, it's part of my ghetto Sharpie.
bro.
When's that 5-minute break though? When's
Now
that
we getting
5-minute
we
break?
ain't
Oh
getting
my
no
God.
5-minute break, my brother.
Nah, bro, no 5-minute break. I
No, I think
gotta
we're
go
gonna
to bed soon.
get more
You've been
bubble
saying,
water.
wait, you've been saying you need
Okay.
to go to bed. Alright,
Now
wait,
we
hold
go
on,
we go
hold on.
next. We go next, okay?
Why do you go to bed so early, bro?
Cause he wakes up at 7, 5.
5.
Why do you wake up at 5? What do you do?
Workout.
What's that?
It was Topo Murray's. Wait.
You should go to Jeffrey Lee's house.
I have my own
Yeah.
gym.
How do you have enough energy to work out,
oh,
shower, and like?
oh,
go
okay,
to
okay, okay. This. Oh, wait, this also, yeah. I'll also write this down. You know how like when you unwind something, it's kind of stressful.
Yeah.
Yo, low-key?
That's where Topo Murray's comes in. It just basically helps release the stress.
Glow and what?
Help unravel.
Yes, not really. All unravel like the song. Wow. Okay, so basically, topoisomerase is just an enzyme that helps relieve the stress when it unwinds. And then after,
it relieves stress.
and then helps it relieves stress
Make the font bigger, Matthew.
yes.
Damn, so demanding.
Please.
Can I run, please?
Yes, yes, yes.
Please.
Go, go, go.
It's an enzyme because it ends in ace.
Can I see yours?
Because it ends in ace.
Yeah. See, anything with it.
That's hella funny, bro.
Anything in that.
Okay,
it
and
is
then
an
the single
enzyme.
strand binding protein just basically, and when after the helical separates into two parental strands, it is the molecules of this single strand binding DNA will bind to the unpaired DNA strands, stabilizing them, and serve as templates. So basically, it just stabilizes DNA parental strand and serves as template. Okay, now we get to this. Okay, good. Everything's handy dandy. We know this stuff, and I'm gonna review it again tomorrow. Yay! Wow. Why do we do this to ourselves? That's what I'm saying.
Yeah, who told us to take AP fucking bio?
Yeah, fuck you, Julian.
Who created
Fuck
AP?
you, Justin. You're the one who said, Julian, you know what you should take when you come to build? You should take AP bio with
Don't
me and
save.
John.
No, fuck you, Julian. You stupid.
Okay.
No.
So what? Okay.
Yeah, the
So what
filler
you gotta know is
class.
that
Yeah.
excision repair. What is
Fuck
that?
you, Julian.
So excision
dick.
repair
Right.
only
What?
happens in. Let me see it. Oh yeah. Excision repair happens when you have a mismatched pair. Happens when you have a mismatched pair. Okay? Mismatched pair. Wow. Basically. No! This is fine.
Don't worry, we know what it is. Mismatched pair.
So
Mismatched
this is the
pair.
process. Okay. You're building your boy. You're building this object, right? You're building your leading strand. But you messed up right here. It's a mismatch. You mess up. And this so what excision repair is a process where it removes that mismatched pair and replaces it with removes the mismatched pair and replaces it. That's basically it. So you gotta remember that excision repair just takes out that mismatched pair, which is right here, takes that out, and then replaces it with the right one. Okay?
Will
And-
we be asked, like, do you think we'll be asked on how it does it?
How does it? I don't know.
How does it do it?
It just. What do you mean? How does it do it? It just takes out the mismatched pair and adds a
Oh.
new one that matches.
Thank you. Okay, got it.
Oh, and also, what you call it? You know? Oh yeah. And it has to be. Okay. I have a question for you guys. Why
shit.
Why does DNA have to be elongated in the 5n to 3n direction? And I give you a hint. Thinking about
Oh,
the
like the carbon thing, right? It's like what carbon number it is.
What do you mean by that? What do you mean carbon number?
Didn't Mr. Wang say something about like what number carbon it was? That's why it's like called, or is that for the name of it?
I didn't
I thought it was the
name of it.
linkage.
I give you a hint. So basically, think about the position of the enzymes and anti-parallel. That's fine. I'll just answer it. So basically, you know what? I'll just create a new one. Yeah, new. Don't save. Okay, so you know that there's a 5n, 3n, 3n, 5n. It has to go in that 5n direction. Move towards here. Because, number one, it's anti-parallel. It has to match up. It just has to match up like that. Because, like, if you go 5n at the end right here and 3n right here, it'll go like this. And you can't bind them together. It's wonky. You can't bind these two things together. But it's also the position of these enzymes. Because remember what I told you how primer starts at the 3n. Okay? Primer starts at the 3n. You can't have the primer start at this part. It does not. That's not how it works. It has to start right here. At the 3n. It can't start at a 5n. Okay? That's the reason. That should be the main reason why it has to start in that way. Anti-parallel in the specific positions of the enzymes. So us humans have this problem, and it's called telomerase. Telomerase is basically about how it's about how you know how us multicellular organisms, unlike prokaryotes, us eukaryotic multicellular organisms, our DNA has an end. So basically, it's just talking about how the presence of telomeres allow Bruh. Okay, this is kind of wonky. The function is to find it. No. Give me a sec. What? Oh.
Can you feel it?
I agree.
Turn it up.
Oh, okay, I found it. So basically it says so basically our main problem in eukaryotic cells is that at the end our DNA Wait.
Oh, I thought it was frozen.
Okay. Basically, us multicellular organisms, we have a problem. Our DNA has to stop. Okay? And that leads to the fact that we have a shortage of DNA molecules over time with rounds of replication. Because every time we replicate, we're replicating DNA, right? But our DNA is not infinite. It's that it has to stop. So when you keep on going and you keep on shortening this DNA, it leads to DNA eroding. So, and that's just a big boy problem. Cause like if our DNA gets corrupted, our whole entire system, our whole genetic info gets disrupted and everything. We basically just die. So what telomerase does, it's an enzyme. No, it's not enzyme. What am I saying? Telomerase is Oh, okay. It's not a gene. It's not DNA. But what it does is it's a sequence that constantly that makes is a sequence of repetitions of adding more nucleotides, and this helps postpone the fate slash doom of us multicellular.
What's a repression?
Oh,
It does say
repetition.
Okay,
Should say the two protective functions.
Wait, what?
The two protective functions of telomeres.
Oh, so so basically, it helps to postpone gene erosion.
Fuck, bro. I got a game Friday too, and I gotta worry about this shit.
Stop crying. Stop crying.
Oh, prevents stagnant cells. Oh.
No, fuck you, Justin. I hope you have work Friday.
Fuck you. Guess what I don't, bitch. Fuck you. Guess what, Julian? I'm getting up.
Wait, Jeffrey, can you tell me what you wrote for the
In
two?
fact, you know what, Justin? I hope
what you
your
call
cut,
it?
your
The
pay
two
gets cut. I hope
the
you
two
get the lost in
benefits
the
of
mail.
like the
I
two
get
reasons
paid
for
$13.25,
telomeres.
you fucking loser, and you jump over fences and cut lawns. Fuck
Like
you.
You
two
make 13
benefits.
an hour.
Yeah, like
working
I have the
ice
first one. It says
$13.25.
host postpone gene erosion.
$13.25.
Wait, can you tell me the other one?
Oh my
Is
god,
that
you got
it?
the
Is it as a
promotion
whole
of 25%.
purpose?
Do you want me to read you the one from the
I had
text?
one
Yeah,
store.
can you read the one for
Only
textile?
one.
Because that's what is. I think that's the whole purpose, though. It just helps postpone
It
gene erosion.
says
Yeah.
prevent the staggered ends of the daughter molecule from activating the cell system
Okay, that's
for
basically
monitoring
that's
DNA
basically
damage. That's
post-
kind of the
That's
same.
the same thing.
There.
Yeah. Okay. So basically, it helps solve the problem of the fact that multicellular organisms don't have infinite DNA, and there can be a shortage over time. There we go. Okay. Basically, multicellular organisms, they don't have an infinite DNA. As they constantly replicate it, it leads to a shortage. That's where telomeres come in, and it's basically a sequence of repetitions of adding more nucleotides in order to postpone gene erosion. Okay, there we go. Dude, why did it take so long? But this is fine. Okay, we're gonna speedrun cell cycle and mitosis.
Oh fuck, that's like the longest shit.
No,
isn't
we
it?
should not speedrun mitosis, bro.
Yeah.
We're gonna go over it tomorrow anyway,
We
so
could,
we
yeah,
might
we,
as well
we
like,
could go
it
over it.
That's
We
what
go
I'm
over
saying.
it at Corey's house tomorrow, right?
Yes.
Just
But-
go over the cell cycle then.
No,
take-
this is
oh,
fine, cause
no,
like
we'll do
I'll
it.
get better at this, and it'll be a lot more efficient tomorrow. That's what I'm saying.
See? And if you gotta leave, then you gotta leave. I'm staying
I-
here.
I have a question.
Yes.
Tommy,
I kind of want to watch the monkey video.
We'll watch it. We'll watch it.
Fuck.
We'll watch.
You're so motherfucker, bro.
Wait,
How long is
Matthew.
it?
Wait, Matthew.
Oh, it's
the
only a minute long.
recording-
Never mind.
Matthew,
Well,
you can,
we're
like
up
send
right
the
now.
recording into like a Discord chat.
yeah,
like
I
a
can
link
probably
to
do
it?
I'll probably put a link to it. Probably just post it. Make it MP4. Make it really long. Okay,
Just send that to Mr. Wang.
that's what I'm saying. Okay, we have cell cycle.
Mr.
and
Wang, is this right? Did I explain everything?
That's what I'm saying. Oh,
Did
okay.
Ju-
This is gonna
Did
be.
Justin
this is
call
gonna
me a
be
bean enough times?
fine. Bro. Oh hell no. Hey yo, we get hit with a suspension. Racially motivated.
They can't
Okay.
do shit. It's at school.
That's true. Okay, okay. You guys
Oh,
think this
really?
cell cycle
Peter-
mitosis might be hard, but really it's a lot easier than DNA replication. Trust, because I reviewed this and I realized it's actually not that bad. Okay, no, what did I? But I'm a buffoon. Yoink. Okay.
Baboon.
Well, we're gonna watch the monkey video, Nathan. We're gonna watch it, okay? Because it's only like
That
1
is
minute
the
long. It's only 1 minute long.
Recessive phenotype for the monkey.
Okay. Okay. There's just some basic vocab. You should know. It's not that bad. Genome equals genetic info. Basically, genome's basically DNA. Genome's basically DNA. What else does it say? Chromosome.
I got a
Basic.
few of those.
Julian, you know if you
extra.
Julian, if you know Julian, did you know
That's
that
funny.
if you have more than the 40, is it 42 or 46?
46.
If you have
More
more,
Yeah, if you have more than 46, you have
special.
that's not good. It is what it is.
You mean it's not good? Do you have
What do you
something
mean?
against people who
No,
have more?
I'm just saying it's not good for it's not good for the
I,
body.
I'll have
like
you
You're
know
not
I have
normal. You're not normal.
47.
packages of DNA, chromosomes, packages of DNA. Oh, and there's one more that I remember. Where is it? Oh yeah, chromatin is basically chromatin. Makes up chromosomes. That's basically, I can't spell, but you get what I mean. Chromosomes. Then stomatic cells are body cells. Stomatic equals body. And what's the other one called? Is it?
Somatic or somatic?
I'll spell it
Oh,
right.
can you define the difference between like let me see what the word for the homo and hetero parts.
Which one?
I
Wait,
forgot
what?
which one.
Say again?
It was the homo and hetero. I forgot what they were.
You mean homo?
I'm gonna go,
Homo is same alleles. Hetero
but
is
like you
different
said, you
alleles.
need to know more than that.
That's literally what you need to know. You're so silly, Tim.
For real, bro. That's what I'm saying.
He said in his lecture, I have it recorded. He said you're gonna need to know more than that.
No,
More than just.
what do you mean? That's not specific, Timothy Butler.
That's
Here,
the definition.
let me try. I'm
though.
gonna
What
go
else
through
do
my
you
voice
know?
memo.
Nah,
The
I'm
lecture?
gonna hit you with a server mute. Nah, I'm joking. So basically, there's
I have no idea
genome,
your
genetic
shit.
info, chromosomes, packages of DNA, chromatin, which makes up chromosomes, stomatic, body cells, gametes, sex cells. Somatic cells are diploid, which is 2N. Gametes are haploid, which is N. Okay? Basically, diploid has all the chromosomes, and haploid has half the chromosomes. Okay? And we need, blur it out, why do we need to do mitosis? That's what I'm saying.
Do it tomorrow.
What do you mean? Just blur out the reason why we do mitosis,
Julian, you
Julian.
can go to sleep if you really need to.
Yeah, Julie,
No,
you
I'll
ain't.
be here.
You don't want your beaner ass here. Wait, no,
Oh
no,
my
no,
God,
no, no, no.
You got no
now,
chill, bro.
now I'm staying.
Yeah.
Yes, yeah.
I'm
help us to
staying
learn.
in
It's
spite
for
of you.
repair and
People
growth.
learn.
Wow, good job, Nathan Slow. That's what I'm saying. Okay.
I just got back from the bathroom.
How was it?
What was it?
Okay.
Damn,
We
don't want to
have that
answer.
down.
Yeah, didn't want to answer
Okay. G1 phase,
I
basically, so we're gonna now go to about mitosis. So G1 phase makes the cell bigger. It doubles the size, basically
I
making it
double your size.
92
Are you?
chromosomes, okay? So in G1 phase, it just makes it bigger. In S phase,
Matthew, guys.
and in S phase,
arm wrestle.
bro, I'm server muted all of you guys.
He gonna bend you over, Julian. He's gonna bend you over.
He's doing it.
No, I'm not. I'm joking, but I get what you.
Yeah, sorry, Matthew.
Okay,
Let him man speak.
so basically, we have G1 phase in all of this, right? And as I said before, G1 phase, it doubles the cell size, hypothetically making it having 92 chromosomes. So after that, after G1 phase, there goes S phase. Now S phase is there for DNA replication. After it replicates DNA, it goes to G2 phase. And G2 phase is the state where it's able to it checks G1 and S phase. Okay, it's like a check. It's like the biggest checkpoint. And then you have M phase.
Wait, what was it? What was the checkpoint?
G2 phase. It just shows what you call it? It just regulates everything, and we can be like, is it good or nah? Okay, and then we have M phase, which officially is the phase where the cell divides. Okay. So we're now gonna go into specifics of prophase and all of that, and
Wait,
it's
Matthew,
really not
Uh,
that. It's not that bad. Okay, I'm gonna draw. Okay.
Matthew,
Wait, what?
Matthew, are you gonna go over the essay questions tonight?
No. Dude, I didn't even know what he said.
Oh, okay.
Have it recorded.
Wow, you're so cool, Timothy. Okay? Okay.
Who
So we
asked
have
Timothy?
prophase. Wow. Prophase is a state of where there's a cell, right? There's a nuclear prophase. The nuclear envelope is still there. But what's important is that and also these are these are the
That's on the
cytosomes.
inside that's important.
The centrosomes. So what's the most important fact about prophase is that inside the nuclear envelope, there's a forming of chromosomes. They're condensing to prepare for. Is it prometaphase? Let me check in the book. Oh yeah, it's called.
Let's let the teacher double check his stuff, guys.
That's what they're doing. That gotta happen. Just so I don't. The teacher is not ever wrong. That is facts. What chapter is this again? Oh, chapter.
I think I made my little
Oh yeah,
my
chapter 12.
prophase look like an egg yolk.
What'd you send me,
An
Tim?
egg yolk.
Oh, you sent.
Send you the what he
Wow,
set
you're
up
so
really quick.
kind. What a fine young gentleman.
Welcome.
Okay, I found it. Oh my god, this is making me cream.
My pencil broke. Fuck.
Okay,
How many times
break
you
this
guys say that, Julian, bruh?
okay, we have prometaphase, right? So prometaphase, then we have prometaphase. Okay, so we know that prophase, the main thing you gotta remember is that there's still the concept of the nuclear envelope, and what's happening in there that chromosomes are forming, right? So at the end of preparing for metaphase, we have this with like this there's slightly a nuclear envelope found, but there's a presence of chromosomes. There's chromosomes. Wow, chromosomes. Okay, and now we have this. We have the prophase, which I'm gonna further reinforce again. It's a stage where there's still the nuclear envelope, but it's starting to form chromosomes. And in prometaphase, chromosomes are now present, but they're starting to prepare itself for metaphase. Okay? So let's create a new one. Hua. Don't save. We got metaphase. Hua.
Damn, you're moving too fast. You're moving like Mr. Wayne.
Bruh.
Don't worry, that's why he's recording.
That's what I'm saying. We have the circle, right? And now we want to line up these chromosomes. And it gets lined up on the metaphase plate, which is this line right here, imaginary line. And metaphase plate, okay? Okay, now they're
Makes sense.
basically half now. They're halved, okay? They're ready to prepare for anaphase. And also, what you gotta remember, this is mitosis, and these chromosomes are identical to each other. And what is identical chromosomes called?
Sister chromatids, right?
Wow, good job, Corey.
Oh,
That's what I'm saying.
The chromatids.
Wow, good
you're,
job,
you're a
Tim.
little late. You're a little late.
I'm lagging, bro. Oh my
You've been like
God.
the lagging strand.
Oh.
I'm joking. I was kind of talking.
Let's stop supporting it.
I like it. Matthew's a good teacher, so you get to fucking respect him, you piece of
Shut
shit.
up, Julian.
What? He's a good teacher. That wasn't a funny
Okay.
joke.
So we know. So it's at a point where at anaphase these chromosomes separate, right? But you gotta remember that since they're identical, they're able to create this identical cell due to the fact that these chromosomes right here, they're the same. They're identical to each other. So after they separate and go to respective ends, it goes into telophase and cytophase, cytokinesis. So it's just so telophase and cytokinesis just talk about how they're at the end now, and those chromosomes are now formed into this one big line, like with a squiggly line of DNA info, respectively right here. Okay, and now they're preparing to become two identical cells. Okay,
I do a snowman.
that's a snowman. You're
Yeah.
right. Okay, new. Don't save. And what you gotta remember is that there's two ways. There's two ways in which the cell can divide. For animal cells, they got the cleavage furrow, the booba. This is the booba. This is the cleavage furrow. Due to the fact that there's no cell wall and there's just pure membrane, it's able to separate. Okay, but in plant cells, they got the cell wall. they already have their stuff replicated and everything, but they don't know what to do. So that's where they have the cell plate, and the cell plate is just basically an additional part where it just becomes one with the cell wall, and then it becomes these two things. Basically, it just becomes with one with the cell wall. So we got the booba, the cleavage furrow for animal cells, and the cell plate for plant cells. Okay. So basically, I'm just gonna speed run this again.
It looks like burger.
Prophase.
Burger and refrigerator.
Ooh.
Also got a burrito to you, huh? Fucking.
I said burger. You...
No, you're thinking about burritos.
Hey Justin, those things Matthew's drawing looking like ramen, huh? You can see chow mein in that, don't you?
That was like a fucking taco to you, bitch, huh? Looking like a green card, huh?
Bet you could probably read this, huh, Justin?
Yeah, I bet you probably could even read it if it was Spanish. Fuck you.
Oh my god, y'all ruthless. Okay. 1, 2, 3. Okay. What's the name of what's the stage of this one? And basically there's
Prophase.
Why? Julaine, tell me why.
A nuclear envelope is present.
Okay, good. What's this one?
That one would be the metaphase, because you can see the metaphase plate.
Facts! What's this one?
Oh, fuck. Anaphase, because they're like separated.
Let's go! Julian knows what's up! Okay.
Matt.
Interphase.
Wow, you got the... Oh, okay, also, can you tell me, why prokaryotes, why prokaryotes think mitosis is a way of reproduction for them?
Wait, can you say that again?
Say that again.
So basically, you got prokaryotes, right? And they also do mitosis, but mitosis for them is reproduction. Why?
Because they're asexual.
Splitting? I don't know.
Wow,
The hairless monkey.
bro, where'd you get that from? Okay, Tim, did you rationally think why would you say something like that?
I didn't think about it. Sorry.
So silly, Tim. Okay, Jonah is right. It's basically
Wait, what did you say?
he says asexual, and they do sex by themselves, because they're single celled. They're single celled.
What a way to describe
How
it.
can the hairless monkey do asexual production? The reason why it is hairless is because of the genetic variation.
Okay, thank you. Okay, so basically, the reason why prokaryotes are called mitosis and reproduction is that they're single celled. There's only one cell. So if they reproduce, if they will do mitosis, and they want to create another generation, they only can create identical. Their generation will always be the same to the parent, and that is called binary
It'll never
fission.
be different.
Wait, what? What, Corey?
It'll never be different, right?
Yeah, it's called binary fission basically. The
Binary fission?
fact that the generation, any generation will be identical to the parent. And as I said before, this is called binary fission, okay? So when you see binary fission in the test, just remember that same prokaryote, mitosis for prokaryote, okay? Oh, and also, it's the children of the parents, the prokaryote parents reproducing is called clones, and clones are literally what it sounds. They're the same, okay? Ooh, we're gonna go about checkpoints in the G phase. This is gonna go really fast.
Oh,
and also, give me a rough estimate of how much does interface take up within the whole thing?
90%.
How much what?
Like 90%.
Nice swag.
I said it first, Jonah.
Hey, I would have said it. I'm just slow.
As I reiterate before, G1 phase is where this is basically G1 phase and all that, interphase and all that. G1 phase, cell becomes bigger. S phase, DNA replicates. G2 phase checks G1 and S phase to make sure if it's correct or not. So we had these two things, okay? They're called growth Yeah, growth factor and density
Ouch.
dependent inhibition. Okay. Inhibition. Okay. So what growth factor does is it's a type. Growth factor is basically a type of protein that gives signals to the cells to let in more cells to divide. So it's basically an internal development. It's inside the cell. Internal. It's an internal development that revolves around replication, replication and division. Okay. So it's a growth factor is reported is inside the cell. It's a protein that gives signals to the cell and that allow more cells to come in to replicate more. Okay. Wow. That density dependent is an external development. So what happens is when the cell creates enough replication and more cells, there is a layer of cells, and this layer of cells is able to stop replication and growth as remember this, as because when you have so many cells, there's ever so much nutrients that can suffice this group of cells. So this layer enables okay, yeah. So we have this. There's this. Oh no. That's fine. I'll show you in the video. There's a layer right here, right? And this layer is a cell. This is a layer of cells, and it's reached its maximum point. It's at its breaking point, and the cell realizes it can't support any more. It can't support any more cells. So that's where it stops, and that's where it stops reproducing, and then it will keep on, and it will start reproducing again if there's no more, if there's like a kind of like a gap between this layer. Okay. Okay. We're done with cell cycle. Oh, we're almost done.
Almost.
Yes.
Shut
There's
up.
one more thing. Okay.
Don't make me come in there and fucking slap your shit, Justin.
We have kinase. Okay, kinase. Kinase is this enzyme that powers the cell cycle, powers mitosis, right? And kinase, in order to power, and in order to a point where it needs to power, it's at S phase to G2 phase, and that's where cycling comes in. Cycling combines with kinase, or known as CDK. CDK.
Kinase?
Yeah, kinase, or known as CDKS. Yeah, CDKS.
Wait. Oh, it's kinase enzyme because ASE. Is it?
Yep.
Yes.
Oh.
Okay, so it's at a state where it's only CDK right here, cause like there's no cycling, right? It goes up, doesn't need it, but it reaches right here. That's where its cycling comes in, and there's a slow amount, cause you only need that much amount for S phase, but then it increases to the point where when a cell divides, at this point CDKS, when a cell divides, they remove cycling out of the way and repeat this process. And this whole process of cycling coming in and coming out is called MPF. This whole process. Okay? And okay, we're gonna briefly talk about tumors. So and cancer. So if cell division is not regulated and gives birth to very deficient cells, these group of cells are called cancer. Okay?
Cancer cells.
Cancer cells.
Mhm.
And basically,
I know about cancer from Breaking Bad.
I know about cancer from my grandpa.
Oh shit.
Oh,
Yeah.
Yeah.
okay, we have the Benign tumor. Okay, Benign tumor. I'll write it out.
Shut up!
Right, the Benign tumor, the Malignant tumor, And we have metastasis. And you got, and all of these are types of like cancers and that stuff. Just all of these types of cancer cells. So the benign tumor is basically is at a concentrated point. It can be as spoken as it can be it can be at a point where it can be regulated. But malignant tumors are basically they're spontaneous. They're able to spread.
Malignant.
Yeah, malignant. Thank you. Malignant tumors are able to spread, making it very dangerous. Well, metastasis tumors are just these cancer cells that pop up randomly, and they're pretty far from each other. Okay. So we're done with that. Okay.
Okay.
Now we're gonna go about meiosis. It's gonna take like 15 minutes.
Bro. Make it 5. Make it 5.
Actually, I can't make it 5. Watch this.
Oh.
Asexual and sexual reproduction. As Jonah said before, asexual is by only one parent, and sexual reproduction is by 2 parents.
All this, I'm gonna get a C on the test.
All this I'm gonna get an F on the test.
That's what
Dude,
I'm
why are you
saying.
shooting so high?
Asexual, so basically I said asexual is 1 parent, sexual is 2 parents. Sexual is known for variation, and the life cycle is basically sperm, egg.
What? Oh.
Right here, they do the thing. Their end. It gets into a fertilized thing. This is fertilization.
Yolk.
Zygote.
Damn, Tim knows.
It's called
Fertilization,
zygote.
and then it makes bebe, and then it rinse and repeats. Okay.
Beh.
Julian.
Don't say it.
Huh?
Booba. Bebe.
Okay, so okay. Eukaryote basically is the pair of chromosomes, and 1 to 22 are autosomal, which is body cells, and the 23 is sex cells. Okay. In meiosis 1, what is it? What was that? Why'd you take a camera?
Maurice, I got his shirt off. Sorry.
Okay. Okay.
He posing.
Okay, we got these.
Yeah, sorry.
It's fine. Okay. Oh, also, note the difference. Homologous pairs are pairs of the same.
Homologous.
Yes, yeah. Homologous is a pair of chromosomes that are from each parent. Okay. While sister chromatids are identical. They're both from the same parent. Okay. So in prophase 1, for meiosis 1, they do the thing called crossing over, right? These pairs of sister chromatids. Wait. Oh yeah.
What's this one called?
This is called meiosis, right? This is prophase
No,
1.
no, this part. Okay.
Prophase 1, okay? These are sister chromatids, right? You gotta remember. Wait, no, these are no. They're home. Oh yeah, wait. Oh yeah, they are Okay. They are homologous pairs. That's what I'm saying. Okay, they are homologous pairs, and this 4 pair thing is called a tetrite. Okay. It's at a point tetrite. Okay. Tetrite. So it's 4 right here. Just remember that when you see prophase 1, you gotta see that there's 4. There's 4 for each thing usually. No, there is 4 for each pair. There could be multiple of these 4 pairs, but as long as there's 4, you know it's prophase or it's meiosis 1. Okay, it's prophase 1. Okay. They do a thing called crossing over, where this gene crosses over to mix with this one, right? And at the end, for meiosis, that goes to metaphase 1. There's these things, right? These chromosomes. They're all right here. The metaphase plate. And all that. But these they do the crossing over. They both sides have genetic variation. And by the law of independent assortment, they can go from each side. They can flip either way. Doesn't matter, okay? And now, they get these chromosomes. They get removed from each other from the metaphase plate, right? And then it forms the thing. Right.
Wait, is that 2 on each side? Matthew?
Yes, there's 2 on each side.
So like, is it like one red and one blue on the left on both sides, and then
Yes.
the other side's
Yeah.
also okay?
You have this thing, right? And it has these. Hey, no, there's only 4. Okay, you have these 2 things, right? 4, and that's telepathy cytokinesis 1. But each thing has variation due to crossing over, right? And after this is the same thing as mitosis. So basically, these 4 things they separate, and then at the end they give 4 offspring. Four possible things, 4 gametes, basically, that each are inherently different. Okay, and I'll probably go further in depth tomorrow.
This is gonna be one of the free response questions. So,
Which one?
what he's talking about right now.
Shit.
So
it's
just keep in mind.
asexual and
Well,
sexual.
fuck.
That's fine. I'll go over it more in this tomorrow. Okay, that's basically all. That's basically meiosis. Yeah, that was really quick. That was,
15 minutes, my ass.
that was 5 minutes.
Shut up, Julian.
Yeah, that's what's up, Corey. You tell him. You tell him, Corey.
Okay, we're gonna watch a monkey video.
What's up, Justin? I saw when Matthew was drawing all the fucking anime shit. It looked like a minion that yo people, huh?
Finally.